Hello students, and welcome to your fourth cell biology lecture. So in this lecture, I'm going to be continuing to talk about the way cells live and reproduce and the way that cells die. So cell cycle, cell division, and programmed cell death. And specifically today, I'm going to be talking about two more sorts of cell division, meiosis and binary fission. And then I'm going to talk about programmed cell death. Okay, so just to remind you what we were talking about last time, we saw that the cell cycle is a series of ordered stages that every cell has to go through as it divides to make two new cells. So we saw that there are phases of G1, S phase, G2, and M. And mitosis is a series of ordered stages which divides the genetic material exactly and accurately between the two new daughter cells. And cytokinesis is the stage of actually splitting the two cells. Okay, so that is the cell cycle and mitosis. So I'm going to start today by looking at an alternative form of dividing the genetic material into new cells, and that is meiosis. So if you remember the video that we watched last time, we asked the question, what is special about the cell division in the cells involved in sexual reproduction? So that is what we're going to be looking at now. And the name for this sort of cell division is meiosis. It's a special type of cell division that's only used for cells that are going to be used for sexual reproduction. So the two purposes of meiosis, the reason why it's different to normal mitosis, is that it allows a stage that shuffles the genetic material inherited from the two parent cells. I've said father and mother here, but it could equally be two microorganisms. And it allows production of cells with half the genetic material, which are known as haploid cells. Haploid, a word meaning half. So when two haploid cells fuse together, two cells with half the normal genetic material, they fuse together and produce a diploid zygote. Okay, so I hope you're understanding the words. Half means half the normal genetic material. In other words, only one copy of each gene. Diploid means two copies of each gene. So we're going from sort of half to two. There's no actual one in the middle there. Okay. So I'm going to start by showing a video which just helps you to visualize what is happening in meiosis.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Okay, so this is a diagram just lying side by side the process of mitosis that we've already looked at and the process of meiosis. And you'll see that they are quite similar in a lot of ways. However, there are differences. So one difference that we saw in the video is that meiosis has a stage called crossing over. So this happens during prophase 1. So for meiosis, we divide the process up into 2 different stages, meiosis 1 and meiosis 2. Okay, and each of these divisions of stages has a set of stages similar to what we saw for mitosis. So prophase, anaphase, etc. Okay, so during prophase 1, the pairs of homologous chromosomes pair up and lie side by side. So make sure that you understand what's meant by homologous chromosomes. Each of our parents passes on 1 copy of a chromosome, and these chromosomes from the father, the paternal homologue, and from the mother, the maternal homologue, are similar. They carry the same genes, but they are not exactly the same. They might carry slightly different forms of similar genes, okay? So the copy from the father and from the mother have the same genes, but might have differences within those genes. Okay, so here is the stage of DNA replication, where we go from having a single copy of the DNA to having 2 copies. The DNA has been replicated. And as I keep saying, we'll look at the details of that next semester. So replicated DNA, 2 copies of the genetic material from the father, 2 copies of the genetic material from the mother. And the chromosomes are now in the familiar X form with sister chromatids. Okay, so each side of the X shape is a sister chromatid. So these homologous chromosomes, each of which has already been replicated or duplicated, line up together. And they line up together in the middle of the cell in a similar way to we saw the chromosomes lining up on the metaphase plate in mitosis. They also line up on a metaphase plate, but with the 2 pairs of homologous chromosomes side by side. And during this lining up process, during prophase 1, crossing over can take place. So I don't know, it's quite small here. We'll look in more detail later, but you can see a joining between the 2 chromosomes here being indicated. Okay, so here are the chromosomes lined up in the middle of the cells, crossing over, taking place and finishing, and then the spindle pulling these two pairs of chromosomes to opposite sides of a cell. Okay, so here in mitosis, we saw the chromosomes being pulled apart directly. So the two duplicated copies were being moved to opposite sides of the cell. Here we're seeing the whole duplicated chromatin with the sister chromatid still held together being pulled to the two sides of the cell. Okay, so here are the details of what's happening in crossing over. So here is the replicated paternal chromosome. Here is the replicated maternal chromosome. So there are actually 4 copies of similar DNA lined up here. Okay, do you see that? And each pair we call sister chromatids, and they should be exact copies of each other, whereas between the maternal and the paternal chromosomes, there will be slight differences. So crossing over then takes place, and parts of a genetic material are moved from one chromosome to the other. So here we see the end of this chromosome. The red one here is joining onto the grey one, and the grey one joining onto the red one. So when these separate again, we will have a red chromosome with a small piece of grey, and a grey chromosome with a small piece of red. And this process happens at many places along the chromosomes. It's not just one crossing over event, as we're illustrating here. We have many crossing over events. Okay, so this is often described as a shuffling process. The genes on each chromosome are exchanged and swapped over, so we no longer have a simple paternal and maternal set of chromosomes. We have a mixture of genes that have come from both mother and father on both chromosomes. Okay, so we've seen that meiosis has 2 different stages, meiosis 1 and meiosis 2. So during meiosis 1, the replicated chromosomes carry out crossing over and are then divided into 2 new cells. So you'll see here illustrated the crossed over part of the chromosome. Then we go on to meiosis 2, which is known as the reduction division. So here we've got 2 cells that each have 1 copy of each chromosome's genetic material. So these then have to be separated again, and we end up with 4 haploid daughter cells. So each of these 4 cells now has just 1 copy of each particular chromosome, but with a mixture of genetic information. So you'll see illustrated here that one has a completely red chromosome, one a completely grey chromosome, one red with some grey, and one grey with some red. And remember that this is just a simplified illustration. In reality, there's a lot more mixing than that. So each of these 4 different chromosomes would be very different to each other. So these are the haploid daughter cells ready to fuse with another haploid daughter cell to produce a new diploid cell. Okay, so this is again is the comparison with mitosis. Okay, So this is what's happening to separate the chromosomes. So this is what we saw last time in mitosis. We saw that the chromosomes are pulled apart by the spindle attaching to the kinetochore, and the two pairs of chromosomes that were joined together in the X shape are pulled apart. Okay, so the attachment here on the centromere, which is illustrated in yellow on these chromosomes, the kinetochore is a protein structure that forms on the centromere and allows the microtubules of the spindle to attach to the chromosome. So this is metaphase, where they're lined up on the metaphase plate. Here is anaphase, where we're seeing the movement of chromosomes. So here are the spindle fibers pulling the chromosomes towards the two spindle poles. Okay, and here is telophase, where the chromosomes are separated into the two new diploid daughter nuclei. Okay, so that's mitosis. I hope you understand what's happening here. So here is the similar process in meiosis. So here we see the four chromatids lined up, okay, the paternal ones in red and the maternal ones in grey. We can see some crossing over going on. Here we've got two crossing over events illustrated. Here are the centromeres and the kinetochores and the spindle attachment, but this time we're not pulling apart the two sister chromatids. Instead, we're pulling apart the two sides of the bivalent chromosome here, okay? So we're actually moving two copies of the genetic material in each direction, but with the crossed over pieces. So this is anaphase 1, okay? So by the time we get to metaphase 2 and the chromosomes are lined up again on a metaphase plate, this time we are going to separate the two chromatids. So again, the adhesions, what was holding the chromosomes together here, the proteins that are holding them together, break down. They're illustrated by little blue lines here, if you can see. These become broken, and that allows the two sister chromatids to separate and to move towards the two spindle poles of the new cell. So by the time we get to telophase 2, we have 4 different haploid daughter nuclei, okay, each with one similar copy of each chromosome. Okay, so the other difference between meiosis and mitosis is that they have an opposite purpose, if you like. So the purpose of mitosis is to make sure that the cells that are produced at the end are genetically identical to the original, that they have exactly the same genetic information. And you'll remember the checkpoints in the cell cycle that make sure that cell division doesn't happen if DNA replication has not correctly happened. Okay, so these two daughter cells should be exactly the same as the parent cell. Meiosis, on the other hand, has the purpose of producing daughter cells which are genetically different to the original. Okay, so genetically different in that it's only got one copy of the chromosome rather than 2, and genetically different in that the gene shuffling of crossing over has taken place. The other way that the cells are different is, remember that when the chromosomes line up on the metaphase plate, they're randomly pulled in the opposite directions. So here we're seeing the paternal or the mostly paternal chromosome being pulled to the left and the mostly maternal chromosome being pulled to the right, but we're only looking at one chromosome here. Remember that human beings have 46 chromosomes, 23 pairs of chromosomes. So if for another chromosome, the maternal one will go to the left and the paternal one will go to the right. So a lot of shuffling, a lot of exchange of genetic material, which is why if two parents have a number of offspring, if we want to think about human beings, me and my husband, we have 4 children, 2 boys and 2 girls, and they are each very different in appearance, in personality, in characteristics. They are 4 different individuals. Okay, so any questions on meiosis? Okay, so I'm going to go on now to think about cell division in prokaryotes. So remember, the big difference between eukaryotes and prokaryotes is no nucleus. But we've seen how important it is for the nucleus to be containing and separating the genetic material in eukaryotes. So prokaryotes, because they have no nucleus, they have a lot less DNA than eukaryotes. The DNA is not packaged with proteins. It's a single ring-shaped molecule for the main bacterial chromosome. And there are sometimes other little pieces of DNA inside the cell as well, things called plasmids, which you'll be talking more about later. Okay, so the main genetic material is the bacterial chromosome, which is a circular chromosome. The other big difference between prokaryotes and eukaryotes is that prokaryotes don't have microtubules inside the cell, therefore they don't have a spindle. On the other hand, the DNA chromosome is actually attached to the cell membrane by a protein anchor. Okay, so bacterial cells reproduce by a process called binary fission. And here is a micrograph of some cells, some E. coli cells, which are going through the process of binary fission. So if you look at this cell, for instance, it's quite short from one end to the other, whereas this cell is a lot longer. This cell, you can see, it's now just about dividing to make 2 new cells. And here are 2 cells which have completed the process of division and are now just 2 cells lying side by side. Okay. Here is quite a nice long one, but is probably about to start dividing. So binary fission is an asexual way of reproducing. The 2 new cells are exactly the same in genetic material as the original cell. They're rather smaller, but then they grow until they're again ready to divide. So the cells are genetically identical, so this can be called a reproductive clone. There is no crossing over, there is no shuffling of genes. Each daughter cell should have exactly the same genes as the parent cell had. Okay, so here is an electron micrograph just showing a bacterial cell, an E. coli cell, in the process of dividing. So you can see that this will be one cell, this will be another cell, and the genetic material is divided between the two cells. Okay, and it's pinching off in the middle here, ready to separate into two separate cells. Okay, so here's just a little animation showing us what's happening. Okay. So there's a bacterial cell, there's its genetic material inside. So the DNA is replicated and to make two identical copies, which are attached to the cell membrane and pulled apart to two ends. The middle of the cell pinches in and the two new cells separate. Should we watch that again? Okay, one bacterial cell, DNA, process of replication and pulling apart of the two chromosomes attached. The cell grows longer, the two copies are pulled apart, the cell pinches in the middle and divides into two, and we now have two new cells of identical genetic material. Okay, so we've got three stages happening here. The first stage is DNA replication, when we make a second copy of the chromosome. The second stage is cell growth or cell elongation, where the cell gets longer and the two chromosome copies which are attached to the membrane are pulled apart. And then the third stage is the actual cell division, where the membrane pinches in the middle and separates the cell contents into two. So each new cell gets one copy of the DNA, of the genetic material, plus it also gets, each also gets half all the other contents of the cell. So any nutrients that are here, any enzymes, everything else that's inside the cell is divided between the two new cells. Okay, so just a slightly more detailed diagram here. This is naming some of the proteins that are involved. Don't worry too much about those names, but this just shows you how the cells are divided by a sort of pinching in of a protein ring in the middle, which separates the two sides of the cell, the two cell membrane sort of pinches them in and pinches them off, and then a new cell wall forms around the outside. Remember, bacterial cells have cell walls, so a new cell wall forms, and the new cell wall has to be provided for this central part where there wasn't any cell wall before. This also shows you a typical time scale of what's happening. It takes about 80 minutes to happen altogether. E. coli under ideal conditions can divide about every 40 minutes, but under most conditions it's more like 80 minutes. Okay, so just to note that binary fission is also the cell division method used by membrane-bound organelles inside eukaryotic cells, especially mitochondria and chloroplasts, the organelles that have their own DNA. So they use binary fission to increase in numbers before cell division. So each new cell gets a good set of cell organelles. Okay, pause for questions. Okay, so we're now moving on to our next topic, which is thinking about cell death. What happens when a cell is worn out or no longer needed? Or what happens if there is something wrong with a cell and it has to be removed before it does any harm? How do cells die? Well, there are two main ways that cells can die. The two important ways that I want to talk about today are necrosis and programmed cell death. So necrosis is unintentional cell death. This is what happens when the cells are damaged. So if you've listened to my lecture about hemoglobin, and when I talked about sickle cell disease and sickle cell hemoglobin, I hope you remember that one of the problems in sickle cell disease is that red blood cells break open, and they do that because long, stiff fibers of hemoglobin are formed inside the cell and break open the cell and release the contents. So that is one example of trauma that might break open cells. Another example is the sort of trauma that we're all familiar with. A person might fall down, for instance, and their flesh, their body becomes bruised and scratched or cut. The cells in that injured part of the body have been damaged by that trauma of falling, or perhaps even a worse accident like a car accident. So if this sort of trauma happens, it tends to break open the cell membrane. Cell membranes are very good at doing their job under normal circumstances, but they're quite easily disrupted. The lipid bilayer can be damaged and the cell membrane can be broken. And if that happens, the contents which are supposed to be inside the cell can spill out and come out of the cell. Okay, we can see here there are a number of breaks in the cell membrane, and the contents are able to come out and be outside the cell. And the release of cell contents outside the cell membrane, is then recognized by the immune system of the body that this is part of, and can trigger immune responses, which can cause things like inflammation. And the inflammation itself, the immune responses themselves, can cause harm to the organism. Some of the cell contents that are being released might be things like digestive enzymes, which can then start to break down other proteins or the outsides of other cells. So the organism can be harmed by this sort of unintentional cell death. This is one of the reasons why if we have an injury, if we have a scratch or a cut, we tend to get swelling at that place, because the immune system is dealing with the problems caused by damaged cells. Okay, so that's accidental or unintentional cell death, a messy process, a harmful process. This is programmed cell death. So this is a much tidier process. No cell contents are released outside cells. It can be described as cells committing suicide, killing themselves. And it's the last part of the normal life cycle of a cell. It's what happens when a cell is no longer needed, when it's no longer needed to carry out its function, or it's no longer able to carry out that function because it's old and worn out. So here you can see a cell, and hopefully you'll notice that the contents here look rather different of the micrographs we've seen before. We've got a number of vacuoles inside the cell, a number of other structures, but notice that the cell membrane is still complete around the outside of the cell. So the contents of the cell are not being released outside. They're breaking down. They're no longer normal cell contents, but they're still enclosed in a membrane. So they're not damaging any of the other cells around. So the name given to this type of programmed cell death is apoptosis. So this is a rather strange word. It comes from a Greek word which means falling off or falling away. So it would be used for leaves falling off a tree. So apoptosis was discovered not so very long ago, about 20 to 25 years ago, and the Nobel Prize in 2002 was awarded to its discoverers. So this word apoptosis was coined at that time. It was invented to describe this process of gentle dying, gentle getting rid of cells. Okay, so here is a cell which has killed itself by apoptosis, and it's been taken up. So it's inside another cell. So this is a phagocytic cell, something like a macrophage, which can engulf other cells. And here is the dead cell that's been taken up. So all the material that's inside this cell, all the amino acids that made up its proteins, all the nucleic acids that made up its genetic material, all its carbohydrates, all its lipids, they can be broken down and recycled, reused. This macrophage can carry out that process of breakdown and recycling. So nothing has been released outside the cell. The inside cell here is still enclosed in its membrane, and it's now also contained in the membrane of the phagocytic cell outside it. Okay, so this little cell is inside the phagocyte. Okay, so here is what happens in apoptosis. First of all, the cytoskeleton breaks down. So remember, the cytoskeleton is the structure of microtubules and actin filaments that gives the cell its shape. So when they are broken down, when they are returned to their protein monomers, the cell loses that shape and it becomes rounder. So here you can see these two cells. Here they're looking relatively normal, and here they're becoming rather rounder. The chromatin inside the nucleus condenses in a similar way to chromatin condensing during cell division, and the nuclear envelope breaks down and lets the DNA of the chromosomes be released into the cytoplasm. So the enzymes which are present in the cytoplasm, which can break up DNA, can work on the DNA and break it up into small pieces. The cell membrane also starts to break up into small bubbles, small pieces, small vesicles. These are called blebs. So here we see a cell in the process of blebbing. It's releasing little spheres of cell contents. Okay, so you can hear it's just starting to do it here. and here it's released lots of blebs of cell contents. So these can be phagocytosed, they can be taken up by other cells, by phagocytes, like this macrophage. So this macrophage has been able to take up a whole cell, but they can also take up small blebs of cells and cell contents. So no cell contents are released outside. It stays wrapped up in cell membrane, in lipid membrane. So it can be taken up by other cells and reused, recycled, with no damage happening to other cells. Okay, so that's what apoptosis is and what we see happening if we observe apoptotic cells through a microscope. So why does apoptosis happen? Well, it can happen as part of normal development in a multicellular organism. So if cells have been put in place during development and then are no longer needed, they can be removed by apoptosis. So here we see this structure, a hand developing. This is actually a mouse's paw, but a human hand, human fingers develop in exactly the same way. So you can see that during this stage of development, the fingers or the digits are not separate. Okay, there is a web of tissue in between the digits. A little bit later, this web has disappeared. And in this picture, a fluorescent label has been used that's marking the cells which are undergoing apoptosis. So you can see that apoptosis is happening in between each of these digits and is allowing them to separate and become separate digits, fingers or toes, or mices. I'm not sure what you call a mouse's separate digits. Okay, so normal development to get rid of cells which were important at one stage of development, but not needed at the next stage of development. So you might be familiar with this sort of idea of metamorphosis. So this is an amphibian, a tadpole, in its swimming, water-dwelling form with a large tail. Here is the same animal at its adult stage where it can live both on land and in water. And by the time it's metamorphosed into the adult form, it loses this tail. Okay, so this is the metamorphosis of a tadpole into a frog. So what happens to this tail? Well, the cells of the tail are removed by apoptosis. So the cells die and are taken up tidally and recycled, and their contents used to allow the growth and development of the adult form of the animal. Apoptosis is also important in getting rid of cells which have been performing their function for some time and have become worn out. Every cell has a limited lifespan. Exactly how long it is depends on the sort of cell, but eventually every cell becomes worn out and needs to be replaced by new cells. So in normal function of a tissue, apoptosis is going on all the time to get rid of cells which have reached the end of their life, to quietly remove them from the tissue that they're part of, and to take their components and recycle them so that they can be replaced by new cells by the process of cell division. So if you think of a normal healthy tissue, perhaps if you think of an organ like the liver in a healthy person needs to stay the same size, but liver cells get worn out and used up relatively quickly. So apoptosis has to be happening to get rid of the worn out cells, and cell growth and cell division has to be happening to make new cells to replace the cells that are removed by apoptosis. So overall, the size of the tissue, the number of cells in that healthy liver stays the same, but there's a turnover in the actual cells by apoptosis removing them and cell division replacing them. And this balance between cell division and apoptosis is important in keeping a tissue to its correct size. If more cell division is happening than apoptosis, we can see growths forming. A tissue can grow abnormally, and this is one of the signs we see in cancer. On the other hand, if there is more apoptosis happening than there is cell division, the tissue would be seen to shrink or atrophy. Okay, so a normal healthy individual has a balance between apoptosis, cell death, and cell division to produce new cells. So we've seen this diagram last time. We've seen that there are a number of checkpoints in the cell cycle which checks to see if the previous part of the cell cycle has happened correctly. So we saw last time that if it has happened correctly, the cell proceeds to the next stage. But what happens if it reaches a checkpoint and the answer is no? If, for instance, if the DNA is not all replicated, or the environment is not favorable, or if all the chromosomes are not attached to the spindle. Well, we saw that the cell can pause and try to put right whatever was wrong, but it's also possible that the thing that's wrong is so bad that the cell can't put it right. So for instance, if the DNA has been replicated, but something has gone wrong, so the DNA has been replicated twice. So if there is too much DNA in the cell can't then easily get rid of the extra. So in that case, the cell wouldn't pass the G2M checkpoint. It wouldn't go on to mitosis. So what it would do would be to go into apoptosis, and then the cell would be destroyed. The mistake that was made would be corrected, and cell division could replace that cell with one that is not damaged. So this is one of the processes that makes sure that cells that have gone wrong aren't allowed to continue to be present, and that most especially are not allowed to divide themselves and pass on that mistake to a new generation of cells. Now, I think that some of you might be thinking, yes, but we know that cell division does go wrong. We know that damaged, unhealthy cells are produced. We call that cancer. So remember, what I am telling you is what is supposed to happen, what happens under normal circumstances, things go wrong. So if we think about cancers forming, the normal mechanism of apoptosis to get rid of damaged and faulty cells is not happening. So that is part of the formation of cancer. Okay, so any questions on why apoptosis is important? Okay, so let's think about how apoptosis actually happens. Let's think about the detailed process of how cells are able to undergo apoptosis. So it starts with a signal that tells the cell that it should go into apoptosis rather than continuing as it is, or even dividing and making new cells. So there are a number of different signals that can tell a cell that it's time to enter apoptosis. So in the example of a developing frog that we saw here, the signal is a hormone that's released from the thyroid of the developing tadpole, travels around its bloodstream, and provides a signal for the cells in its tail to start breaking down, to start the process of apoptosis. So developmental apoptosis, a hormone signal sent at the right time, at the right stage of development. I've got a video for you now, which just helps you to see what's going on in this process, and then we'll look at the details of the process. So here we see just one cell which is unhealthy, diseased, amongst some normal cells. And here is one of the cells of the immune system, a killer T cell. Okay, so now we're looking what's happening between these two cells. So the killer T cell is recognizing that there is something unhealthy about this diseased cell. And it's releasing a signal called here a death ligand, which is being received by receptors on the surface of the unhealthy cell. So the immune system cell has told the cell that it's unhealthy and needs to destroy itself. So here we see a series of interactions between proteins. Okay, proteins interacting together, and now we've got an activated caspase protein. So this caspase is now able to act as an enzyme and activate other caspase proteins. So this is a cascade process. So that caspase is now feeding other proteins. Okay, so a cascade, one thing causing another thing to happen. Here we see the mitochondria, And these signals are reaching the mitochondria and changing the mitochondria so that cytochrome C is being released outside the mitochondria. The cytochrome C can bind to another protein and activate it, and that starts to form a structure. Okay. So this is a structure called the apoptosome, activating yet another enzyme, another caspase. Many molecules of the other caspase now becoming activated, and now they're going off to start cutting up the cytoskeleton, breaking the actin of the cytoskeleton into its separate subunits. So we're back looking at the cell from the outside now. So we can see that because the cytoskeleton has been broken, the cell loses its normal shape. And now we're starting to see the blebbing happening, the membrane vesicles being released. The cell is breaking up, and each of these little blebs, little bubbles of cell content surrounded by membrane, can be taken up by other cells and their contents reused. Okay, so let's go through that process again. So a number of different signals can be released from the cell. They can either come from the outside of the cell. So we've seen the example of the hormone being received for the metamorphosis of a tadpole to a frog. And we saw in the video a signal coming from an immune system cell, which has recognized that there is something wrong with a particular cell, and the immune system cell has sent a signal to the cell to go into apoptosis. The signal can also come from inside the cell itself. So if the cell is starved of oxygen and has become damaged because it's not able to carry out its oxygen-dependent respiration reactions correctly, this affects the mitochondria, which are of course involved in respiration, and causes the release of cytochrome C and sends the cell into apoptosis from inside the cell. So this is called the intrinsic pathway. So a number of different signals all interacting together to tell the cell that it's time to go into apoptosis, that it's time to destroy itself. Okay, so what happens once the cell has received these signals and becomes committed to apoptosis? Well, we have an enzyme cascade. I think that you might have already learned about the blood clotting cascade in physiology. So the idea of a cascade is that a signal changes one molecule. That one molecule can then change a number of molecules. Each of that number of molecules can change many more molecules. So at each stage, there is an amplification. The signal gets stronger and stronger at each stage. So in apoptosis, the cascade is the caspase cascade. And one molecule of an initiated caspase can become activated by receiving the original signal. This can then go on to activate many other molecules of executioner caspases. Okay, so just as the name implies, these are responsible for killing the cell. So caspases belonged to the class of enzymes called proteolytic enzymes. In other words, they are enzymes which can cut protein molecules at specific places. So the initiator caspase cuts the executioner caspases, and this cutting converts the protein structure of these enzymes from an inactive form to an active form. So each of these executioner caspases is also a proteolytic enzyme, and each can then cut other enzymes and proteins. Okay, so here are proteins being cut up by the executioner caspases, the proteins in the cytosol, the cytoplasm of the cell. Other types can cut up other proteins, such as the proteins that make up the nuclear lamina. Okay, so remember when we looked at the nucleus, we saw that there is a layer of fibers just under the nuclear membrane called the nuclear lamina. And if these proteins are cut up and this layer of lamina is destroyed, the nuclear membrane stops being a membrane right round the nucleus and instead dissolves in exactly the same way that we saw in mitosis and becomes little blebs, little vesicles of membrane. Okay, so executioner caspases cutting up the nuclear lamin, breaking the nuclear membrane up, and therefore releasing the chromatin, the DNA, into the cell. Other executioner caspases can then cut the DNA itself into small pieces. Okay, no, I've got two of these. Okay, so what I've got here, let's just pause this if I can. Okay, so here is a series of micrographs of a living cell which are going through a process of apoptosis. So this is actually a mouse cell, but where cell division has gone very wrong. So it's ended up with one cell with many nuclei inside it. So let's, can I just pause it and see this? Sorry, it's a bit hard to pause it at just the right place. Go away. Okay, so let's try again. Stop it. Okay, so trying to format it. I apologize for a technical problem. Okay, so I hope that you're seeing that here is a cell that's got lots of nuclei inside it. It's just the same set of pictures being looped round and round. So here, the cell is wrong. And very quickly, it starts to shrink. So here is a multinuclear cell, and then suddenly it shrinks and starts to undergo apoptosis. Here are the separate blobs. And this video is a set of photographs taken of the cell over a period of about 12 hours. The cell can't divide properly because it's got too many nuclei. It can't undergo normal cell division. So it goes into apoptosis and becomes destroyed. Okay, and you can see the sort of extrusions of membrane on the outside, the blebs forming, and finally, each of those blebs will be taken up by apoptosis. You can see that it happens quite quickly. Once the cell goes into apoptosis, it very quickly shrinks there. That's as the cytoskeleton is broken up, and then the blebbing process. Okay, so there's still a lot we don't know about apoptosis. A lot of the details of the process, a lot of the details of the signals that are involved are still being found out, are still being worked out. So it's a lot of research going on into apoptosis because, of course, there is a connection with diseases such as cancer, where we want to encourage processes that will get rid of cells that are wrong. If I bring us to current news, when a virus like the COVID virus enters and invades a cell, one of the ways that an infection like that can be dealt with is by killing the cells that have taken up that virus and stopping the virus reproducing. So again, apoptosis is important in controlling viral diseases such as COVID. So a lot of research into drugs which encourage apoptosis, which help targeted apoptosis in the correct cells and make sure that it doesn't happen in cells that shouldn't be being destroyed. Okay, pause for questions. And that's all I have for you today. If you want to read more about these things, remember Albert's Molecular Biology of the Cell is a very useful book. I hope you know how to get soft copies of it. Chapter 17 is about the cell cycle, so that covers mitosis and also meiosis and cell division. And chapter 18 is called Cell Death and covers apoptosis. Okay, so that's all for today, and another lecture for next time.